Chapter 2, Section 3. Let me adjust this. Chapter 2, Section 3, the cell wall. Now, you need a cell wall, especially for these single-cell organisms that, for all intents and purposes, would be squishy. If they didn't have something solid, like this table or that wall behind my camera, to maintain the shape of that cell, cocci, bacilli, whatever. You have to keep in mind that bacterial cells are at the mercy of their environment, hypotonic, hypertonic, whatever. High salt, low salt, high mineral, low mineral will cause water to move into or out of the cell. Cell swells up too much, bursts. Cell shrinks too much because all the water is leaving it, collapses, cell dies. So by having a cell wall, it will allow that cell to have some rigidity, counter osmotic pressure, so it can only swell up so much. Cell wall holds it there. It can only shrink so much. The cell wall's anchored to this cytoplasmic membrane and won't let it shrink anymore. And as I said, because of this cell wall, the majority of bacteria are going to have some form of a cell wall. It's just, is it thick or is it very thin? Gram positive, gram negative. The gram positives, gram negative, will have the cytoplasmic membrane and then a thick cell wall. Remember, that thick cell wall is what will retain the crystal violet / iodine crystals. The thin cell wall, cytoplasmic membrane, a thin cell wall because it's gram negative, and then there's going to be an outer membrane. Between the dual membranes and the thin cell wall, the gram negatives like E. coli, Salmonella, Proteus, or whatnot will be able to maintain their shape. Gram positive, gram negative. Gram positive, look, that nice, thick Pepto-Glycan layer. The gram negatives, well, there's the dual membranes, and then that thin line right there is the thin cell wall of gram negatives. And when you look, if you look in the textbook, you can see that they have taken electron micrograph pictures of these to show that thick, what they refer to call Pepto-Glycan layer, that thick cell wall of gram negatives. And then you can barely make it out, the line, if you look at this, you can barely make out the line in gram negatives, but you can clearly see dual membranes. That cell wall of bacteria, it's composed of what is referred to as Pepto-Glycan. I just said that on the last slide. And it should have caused some warning bells to go up. I've never heard that one before. What is this Pepto-Glycan? Well, rule of thumb for all science courses. If it's a new word and you don't know what it means, break it into its parts. Peptido means protein. Glycans is another way of saying sugars. So these are proteins and sugars that are going to be linked together to make a somewhat rigid cell wall. They're found in bacteria. Okay, this Pepto-Glycan is found in bacteria. We're going to see in a few slides, I can't remember if this section or the next, that there are a few that don't have cell walls out there. There's exceptions to every rule. Pepto-Glycan, not going to be found in archaea, not going to be found in us eukaryotes. The peptide portion of this Pepto-Glycan is actually a tetrapeptide, tetra 4. So it's 4 amino acids. You're going to have a sugar backbone that's made up of 2 sugars, N-acetyl glucosamine and acetyl muramic acid. you'll see the acronym for N-acetylglucosamine, N-acetylmuramic acid, used in many textbooks, many papers, primary literature. The acronym for these is NAG, N-A-G, and NAM, N-A-M. NAG, NAM, the two sugars, and they're basically going to be NAG, NAM, NAG, NAM, NAG, NAM, just linked together in a polymer. The tetrapeptide, well, that can vary species to species a bit, but most part alanine, L-alanine, D-alanine, glutamic acid, lysine are going to be some of the main players. So when you look at it, okay, N-acetylglucosamine, N-acetylmuramic acid, based on a 6-sided sugar, glucose, beta 1,4 linkage. Beta just means, which direction is the hydroxyl pointing up here on carbon 1. Carbon 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. So it's 1's linked to 4, hence the name beta 1,4. Remember, this is a glycosidic linkage whenever you link 2 or more sugars together in a polysaccharide. And here's the N-acetyl group, N-acetyl, N-acetyl, and then our 1, 2, 3, 4 amino acids. You're going to have this long chain of NAG-NAMs, and these amino acids are going to be interspersed, and it's going to link neighboring chains together, because one chain is floppy. 2 or more, wherever one chain has to move, well, you got to move a few thousand this way also. And this one moves, you got to move 1,000 that way. So it gets harder to move things when they're cross-linked by that tetrapeptide. These Pepto-Glycan strands run parallel. So if this is the cell, they're going to run like this, wrapping around the cell wall, making the cell wall, cross-linked with peptide bonds, that tetrapeptide. And what's interesting is you're going to get layers of these NAG-NAM strands. You look here, this reddish brown that you see right there and there and there, those are long polymers of NAG-NAM. And then the blue is the tetrapeptide linking them together. The gram-positive cell wall is going to have multiple layers of these. NAGs and NAMs, sheets. Think of it almost like sheets is the way it was taught to me. You look at a sheet of paper, and it turns out that sheet of paper is made of fibers. Okay, kind of think like our polymer of NAG-NAM. Since they're all cross-linked together, wherever this one moves, all the rest have to move too. But the problem is, it can bend, it can curl. So what do? Well, you just start layering. You start layering them 15 or more layers thick. And then you anchor the layers together. How are you going to anchor them together? Well, you're going to use a thing known as teichoic acids. One layer, bendy, bendy, bendy. 15 or more layers on top. Individually, they're bendy-ish unless you anchor them all together. Kind of like stapling them all together. That's what this teichoic acid is all about. The teichoic acid anchors the layers of Pepto-Glycan together. And then you have a special type of teichoic acid known as lipoteichoic acid that not only does it go through all the layers of peptidoglycan, it then inserts into the cytoplasmic membrane, anchoring the cytoplasmic membrane to the cell wall. Now this peptidoglycan, it's not like, a cinder block wall that, can't be destroyed. Turns out it can be relatively, easily damaged using enzymes such as lysozyme. Lysozyme is an enzyme we have in our tears, as an example, that goes through and just starts cutting. It starts randomly cutting that glycosidic linkage between sugars. So instead of a nice, you get this very chaotic, sugars attached to each other, and they're not firm. Thus, the cell can now swell and collapse and will ultimately pop. Interesting enough, one of our original best loved, best used antibiotics targets cell wall synthesis. Penicillin goes and interferes with that cross-linking by the tetrapeptides. So you'll get all of these NAG-NAM polymers, but since they can't link together, they're all flexy-flexy. The cell wall has the rigidity of a cooked noodle. So layers and layers and layers. Anchored together by teichoic acid, and then joined together by teichoic acid, and then anchored to the cytoplasmic membrane by lipoteichoic acid. We're going to take a side jaunt here, and we're going to talk about some pseudo gram-positives here, in the sense, the thick cell wall thing. So the peptidoglycan of bacteria is not going to be found in our friends archaea. Archaea use other means to maintain shape. One of them happens to be that S layer I talked about before. But the main thing is replacing one of the sugars found in prokaryote peptidoglycan with something that is known as pseudomurine. So now you have an acetylglucosamine, NAG, and you have an acetyltalosaminic acid. Something similar to NAM, but really not. A beta-1,3 glycosidic linkage instead of our beta-1,4. Amino acids are all going to be different. And this muramic acid that we're seeing here, that's making up our cell walls, resistant to lysozyme, resistant to penicillin. So when you look at these guys, here's our friend NAG, but something new here. And look at this. The sugar here has been flipped. So instead of both of them being oriented so the N-acetyl groups are pointed down, this one's flipped. So now, 1, 2, 3. So carbon 1, carbon 3. You have a cross-linking peptides. It's not the tetrapeptide like we saw before. This is going to give the cell wall of these archaea a completely different